Ladies and gentlemen, please welcome John Smeraglia to the stage.
Good morning, folks. How's everybody? I think they're going to work on these slides in a minute. Hopefully, I don't have to just without slides. But my talk will be more about biomarker strategy. It's not going to go into the technical depth that we just heard. It really rather focusing on context of use, our assays, and where we need to be interfacing with a range of different partners within pharma. Typically thinking of clinical pharmacologists, translational medicine colleagues, the clinical development program. So it's going to focus primarily on that. Right, let me see if I got this working then. Okay, so I gave a little introduction. This slide, really the key message here is that actually biomarkers are used throughout the development paradigm. It's really we're focusing on novel targets coming into the portfolio, what that specifically means relevant to our target, and our target validation is really fundamentally important. And then we break that down further in terms of looking at dose and getting into the clinical trials. So which dose is more appropriate? How do we determine that? Again, another very important tool being the biomarker to help us to understand our exposure versus our response. And of course, there's the PD and the clinical, later stage clinical development aspects. And this goes a little bit deeper into the same point, and that is that effectively we're really working and thinking about our biomarker strategies right in the beginning. Target comes into the portfolio. We're focusing on MOA. We're trying to understand our target and understanding and working with our research colleagues early on. At this stage, it's far more a thinking exercise than it is a analytical tools and building analytical tools. But of course, as we go into preclinical studies, developing PK/PD studies to determine where we enter as far as dose is concerned, there's a lot of translational discussions and translational understanding of how we can then define what dose we go into. And I will talk a little bit about a safety-related translational biomarker later on. Clinical, when we get into clinic, of course, so now we're really getting into the meat of it to find we're talking about dose, PK/PD models. Are we verifying our PK/PD model? Are we needing to modify our PK/PD model? Further on from there, then we get into the far more towards the regulatory aspects of patient selection, patient stratification, impacts to the label, and of course, in some cases, going all the way out to building diagnostic tools. When we talk about a biomarker strategy, we talk about these elements of it, and that's to say, ensure that we have, PK at the site of action. Certainly if we can get that, and that's not always the case. If it's a circulating endpoint, then we can do that, but oftentimes it's not. Additionally, we look at target occupancy, target engagement, biological effect, or effectively PD in circulation oftentimes, looking at clinical response, and ideally looking longitudinally at that clinical response, and seeing actually the development of disease and the effect on the biomarker itself. And throughout that, you have the safety components of that as well. So there are multi-factors in developing a biomarker strategy, and we really do need the input, thoughts, perspectives of our discovery colleagues, our clinical pharmacologists, our translational medicine colleagues and clinicians to actually develop that biomarker strategy. It's not just from an analytical perspective, but far more a broad spectrum of different expertise that's built into it. But once you've developed all that, once you get some of that understanding, then we really start thinking about the assay itself. And I think that's a critical point, and probably just to accentuate it. Too often, it's because we can. There's an assay that might be able to do something. We're trying to focus the thinking and change the thinking around what is the context of use? Why do we need this biomarker? Do we really need it? And sometimes we do, and sometimes we don't. Or perhaps we need it to a point, and ultimately we do it in preclinical species, but only carry it forward to a limited way in the clinical studies. Very different from building up towards diagnostic, because almost the pyramid is shifted the other way. But I think that discussion, that understanding of the biology is fundamental to building an appropriate assay strategy. Once you have that, you can develop an assay strategy. Without that, it's very difficult to develop an assay strategy that's going to give you maximum value for the time and energy you put into it. So this little slide here is to speak of, again, that, knowing the context of use, defining those assay criteria. Do we have a down-regulated biomarker? Do we have an up-regulated biomarker? What relative sensitivity do we need? And that, of course, is going to then go towards the technology platforms which we decide to investigate, utilize. develop those methodologies that are robust, specific, help us to truly be able to answer that context of use, ultimately validating those assays. And that's, as we heard, quite an extensive exercise. But it's fair to say that sometimes you're going back from there and going, oh, that validation hasn't worked, is not going to answer the question appropriately. We're going to have to go back and think of a different technology sometimes. And then only after all that, you get to actually measuring a sample and testing your hypothesis. And depending on what you get out of that, you can obviously go right back again and defining your context of use over again. So there's a lot of feedback loops within this process. And I wish it was easy, but I'd find no simple way of doing it, sadly. This is a busy slide. I'm not going to spend too much time on it, but the key message of this is that assay and developing the assay, to look at it, and when you think about what the robustness testing you want to do, determine what it's going to be used for. If you're going to be used in vitro, preclinically, do you really want to spend all the time, energy, and effort to develop an assay that actually is ready for clinical utility, and perhaps not. So the focus there is very much, on this slide, is very much as understanding the question, develop those methods, but then characterize them, validate them to the extent that it will answer the question, as opposed to one size fits all. A little bit of change away from the analytics, but again, from a biomarker perspective, we need to be looking and thinking a little bit differently. We need to think, what is that best sample? How do we collect that sample? What kind of technologies I mentioned before? Too often with a number of different proteins, there are a range of different modification, post-translational changes that occur. And if we actually are not cognizant of that, we find ourselves in a position where we've collected a sample, get very ambiguous data at the end of it, and then you're in a situation of trying to interpret what effectively could have been avoided by collecting that sample in a particular way and avoiding all that up front. I think people have seen this before. It just talks about technologies. I won't go too much into this, but it's fair to say that different technologies will give you different outputs. And this is a further breakdown of that, a little more specifics. Again, I think the key message I wanted to provide here is we utilize a toolbox approach. It's not to say that because we are experienced immunoassay scientists or mass spectroscopists or flow cytometry folks, we use that technology. It's the question. It's what is the context of use. That defines the assay and the test system you want to test it in. You've seen some of this already. So again, I won't spend too much time on this, but different, and I have not even gotten into the more high sensitivity platforms and more of the same. The point again being, once you know what the question is, then go to the technology to actually answer that question. If it's a sensitivity issue, if it's a selectivity or specificity issue, there's a technology that you're likely to be able to answer it, but it takes the work to develop those assays. This is one slide to try and explain quite a complex aspect, and that is that there's so many different degrees of variability, so many different angles where variability comes in. I talked about the sample and collecting the sample appropriately, but then this is obviously speak of you're going to have between subject variability. You do need to find that from a range perspective so you can determine what shift you may or may not see. Additionally, you need to be thinking about within subject and the variability of within subjects and diurnal variations or other aspects that will actually mean that you'll have variability within the subject itself. And of course, the analytical variability. And again, and there's the CPATH papers that speak to a great deal of this. This is one slide to try a very important message, and that is there are a number of different aspects of variability, and you do need to characterize and understand that for your assay to be able to interpret your data appropriately. Okay, that was a quick run through. I'm sorry for the pace. Hopefully it makes reasonable sense. What I've decided to do is to just give you one example and change it a little bit, and that is go into the preclinical environment in terms of what that may or may not tell us going forward clinically. And this is all about TTR assessment. So simple slide about the biology effectively, talk about barriers within the brain, blood-brain barrier, and the role of different proteins and different aspects of getting drugs and or biomarkers into the CSF. So TTR transthyretin its function is really to transfer thyroxine and retinol into the brain, across the blood-brain barrier. There's some specifics there, but fundamentally, the effect of this barrier really does have a significant effect on material, proteins and drug entering the brain. And in this case, we had to do it because of the concerns over vacuole formation with a anti-pegylated molecule. And that's really why we decided to take it forward. So what are those considerations? We need to understand the study design, the source of the matrix, what we needed in terms of plasma and CSF to understand the crossover from the blood-brain barrier, the sample size, what kind of samples we might get, how difficult they would be to collect, would the integrity of the sample be compromised, also the frequency of sampling, and of course the stability. We didn't have a whole lot of knowledge on stability at that particular point. And as I spoke earlier, there's inherent variability in some of these biomarkers. Further on from that, from a method availability perspective, we looked at a range of different methods that are out there. There certainly were methods that are out there, different performance characteristics. Ultimately, we decided to develop an assay internally that would give us the sensitivity that we felt we required. So just to note that there is a clinical chemistry analyzer, but it was about the sensitivity necessary, and because the levels we expect to be lower than that range, we needed to develop something internally. We decided to use a mass spec approach and use immunocapture mass spec assay, which would actually get us to the levels we thought we'd see, at least within the toxicology study that we were running. So again, we looked at all the needs of the study, but actually we had to switch platforms. It would have been great if we could just use the autoanalyzer, but unfortunately we couldn't in this case. And some data. Again, this is from the monkey study that we ran. Effectively what we're seeing here, there's some small magnitude shifts, but we have our control group there at the top, and then you have the 30 mg and the 100 mg dose groups. And as you can see, there really isn't much variation between those different dose groups, which at least is telling us that effectively we're acting similar to control group. And that, of course, means that we're not seeing a significant effect based on the blood-brain barrier effect. Additionally, I just put in there as a parenthetical the thought that actually we're seeing levels that are not wholly dissimilar to some of the human levels. And that is a quick run through to try and help out with time. But before I go off, I just want to make sure and say thank you to a range of different people. And there are so many folks that help out here. I particularly wanted to point out a couple of my colleagues over at UCB, as well as our non-safety colleagues. Thank you.